Cell division and the cell cycle. What is cell cycle? Here is a step-by-step instruction manual that a cell follows every time it needs to divide. It has 4 main phases. G1 phase, the cell grows and decides whether to divide. S phase, DNA is copied. G2 phase, the cell prepares for division. M phase, the cell actually splits into 2. G1, S, and G2 together are called interphase. And here's an important one, G0. This is when a cell exits the cycle and just chills. It's quiescent, not dividing, but still active. Growth factors can wake these cells back up. The whole cycle takes a minimum of 12 hours, but in adult tissues, it can be much longer. But always remember, not all cells divide the same way. Now let's talk about the 3 main populations. Stem cells are the parent cells. They're long-lived, self-renewing, and divide infrequently. A classic example? Hematopoietic stem cells in your bone marrow. Transit-amplifying cells are their offspring. They divide rapidly, but they're on a one-way street. They're destined to differentiate and die. No going back. Differentiated cells are the specialists, neurons, muscle cells, and others. Some, like neurons, permanently lose the ability to divide. So the body's strategy is, stem cells maintain the stock, transit cells do the heavy lifting, and differentiated cells do the actual work. Here's something fascinating, not all tissues divide at the same speed. The intestinal crypts are one of the fastest dividing sites in the body. Your gut lining is constantly being replaced. After a skin wound, cell division spikes, then returns to normal once healing is done. The body is constantly self-regulating. And the key rule here, division rate is tightly coupled to demand. When this coupling breaks down, cells either fail to replace themselves, or they overgrow. And when they overgrow? That's called a neoplasm, a tumor. Quick note, G1 is the most variable phase. It can be less than 2 hours in fast-dividing cells, or over 100 hours in slow ones. Let's look at this diagram, figure 1.22 from Gray's Anatomy. You can see the cycle moving clockwise. At each stage, a different cyclin CDK complex is active. G1 phase, cyclin D plus CDK4. G1 to S, cyclin E plus CDK2. S phase, cyclin A plus CDK2. G2, cyclin A plus CDK1. Into mitosis, cyclin B plus CDK1. You'll also notice checkpoint 1 and checkpoint 2, those are the quality control gates. We'll come back to those. So what are cyclins and CDKs? CDKs are cyclin-dependent kinases, are like engines. They do the work, but only when a cyclin plugs in and activates them. Different cyclins appear at different phases, like keys that unlock each stage of the cycle. And here's the clever part, once a phase is done, the cyclin is destroyed. How? The cyclin gets tagged with a small protein called ubiquitin, by an enzyme called the 3-ubiquitin ligase. This tag signals the 26S proteasome to chop it up. No cyclin, CDK goes inactive, phase ends. Clean, precise, and irreversible. Now let's talk about the checkpoints. Think of them as security guards at the cell cycle gates. Checkpoint 1, G1 to S. Cyclin D binds CDK4, signals the cell to prepare for DNA synthesis. Then SPF, that's S-phase promoting factor, which is cyclin A bound to CDK2, enters the nucleus and starts DNA replication. Checkpoint 2, G2 to M. MPF, M-phase promoting factor, which is cyclin B bound to CDK1, triggers mitosis. It causes chromosome condensation, nuclear envelope breakdown, and spindle assembly. And there's also a DNA damage checkpoint at G2. If radiation or chemicals have damaged your DNA, this checkpoint delays division to allow for repairs. Lose this checkpoint, and damaged DNA gets copied. That's when cells can become cancerous. Let's walk through mitosis step by step. Entry: CDK1 activated by cyclin A and B. Chromosomes condense, nuclear envelope breaks down. Metaphase: MPF activates APC or C, the anaphase promoting complex. Anaphase: APC or C destroys cohesins, the proteins holding sister chromatids together, and they separate to opposite poles. Exit. B cyclins are tagged with ubiquitin and destroyed by the 26S proteasome. CDK1 switches off. New G1 phase. D cyclins bind CDK4, and the whole cycle starts fresh in each daughter cell. Now for arguably the most important protein in all of cancer biology, p53. p53 is often called the guardian of the genome. Here's why. When DNA is damaged, p53 levels rise. It binds to DNA and activates the p21 gene. p21 then blocks CDK2, and the entire S-phase engine stops. The cell pauses. DNA gets repaired. Then division resumes. But what if p53 is mutated? It can't bind DNA. p21 is never made. CDK2 stays active. Damaged DNA gets replicated. The cell becomes oncogenic. It starts behaving like a cancer cell. This is why TP53 is mutated in roughly 50% of all human cancers. And in Li-Fraumeni syndrome, the TP53 mutation is inherited. These patients develop multiple cancers, often at very young ages. Now let's give credit to the scientists behind this knowledge. Muller and Vuzden, 2013, published a landmark paper in Nature Cell Biology showing that mutant p53 doesn't just lose its protective function. It gains new oncogenic powers. It actively promotes invasion and metastasis. Chen et al., 2022, in Cell Death and Disease, gave us a comprehensive review of all the ways mutant p53 drives cancer, and what we can do about it therapeutically. These two papers are essential reading if you want to go deeper into cancer biology. Let's bring it all together. Your key takeaways. The cell cycle is G1, S, G2, M, an ordered, tightly regulated sequence. Cyclins plus CDKs drive each phase, different pairs for different stages. Phase transitions depend on ubiquitin-mediated proteolysis via the 26S proteasome. Checkpoints verify readiness before each major transition. p53 is the master braking system. It halts the cycle when DNA is damaged. Mutant p53 means no brakes, which leads to cancer. Li-Fraumeni syndrome is the clinical consequence of inherited TP53 mutation. And that's a wrap on cell division and the cell cycle. If this helped you understand the topic better, please hit the like button. It really helps the channel grow. Subscribe to Gray's Gyan so you don't miss our next video. Drop your questions in the comments. I read every single one. Until next time, keep learning, keep growing.